So I'd like to thank Twist for inviting me to talk at this virtual seminar and for the chance to talk to you about some of the more fundamental work that we've been doing in protein engineering that has nevertheless been underpinned and enabled by synthetic biology, particularly the use of gene synthesis. So this talk has a rather grandiose title of Travelling Back in Time with Synthetic Biology, and it will focus on a lot of the work that we've been doing using an approach called ancestral protein reconstruction, which allows us to basically reconstruct ancestral protein sequences and try to understand how certain functional transitions have occurred. So to give you a bit of background about what we do, my lab is really focused on protein evolution and engineering. We do a lot of computational work with protein evolution and bioinformatics, and my training really began in the area of protein function and protein structure with quite traditional techniques like enzymology, thermodynamics, X-ray crystallography, and increasingly nowadays we're using molecular dynamic simulations to try to understand how proteins move and the impact that this has on their function. But the point of all of this work is really to underpin our work in engineering, where we try to apply what we've learned from this fundamental stuff to generate new enzymes and new proteins for medicine and industry. So a lot of this talk will focus on proteins that have been generated using ancestral protein reconstruction. So I thought I'd give a bit of an overview of this technique. So it starts out, you generate a multiple sequence alignment from extant sequences that are homologous. You have to be very careful of various artifacts that can arise through doing this, but if you do a good enough job, then you can infer a phylogeny or a phylogenetic tree shown here, and the ancestral nodes. At these ancestral nodes, you can infer the sequences of these putative ancestors. These ancestors then can comprise or constitute an evolutionary trajectory where you should, hopefully, if everything works properly, be able to look at the transition from an old activity or an ancestral activity or property through to a new activity or property. The techniques become more visible because a lot of these ancestral proteins have got properties that are valued by protein engineers, things like increased stability. But for us, it's really interesting because it allows us to identify and rationalize function shifting substitutions and mutations. And really highlight the drivers of evolution, things like protein dynamics, epistatic interactions between mutations, and things like this. So this approach, though, is critically dependent on gene synthesis, although it was first postulated decades ago by Pauling and Zucker Kandel. It's really inexpensive gene synthesis, which allows us to test tens to hundreds of ancestors for every experiment, that has sort of taken this to a different place, and that rather than having to hypothesize and guess what the ancestral functions of these proteins might have been, we can now actually make them and have them tested ourselves. So in terms of the talk outline, I'll just go through 3 recent papers looking at the evolution of binding specificity, the emergence of enzyme activity, and how the way proteins evolve to move is important for both of these things. So the talk focuses, all 3 papers are focused on the solute binding protein superfamily, and these undergo an open to closed conformational transition. They exist in equilibrium between open and closed states. When the ligand binds, they will stabilize in the closed form. They're basically ubiquitous. They're found commonly in bacteria, for instance, in the periplasm as parts of ABC transporters. They will bind the ligand. The closed state will then bind to the transporter or a receptor for chemotaxis. In eukaryotes, they've become fused with membrane proteins and constitute the ligand binding domain of menoreceptors, where this conformational change is transmitted through the membrane protein and signaling, for example, with the AMPA receptor. So the first project was looking at the evolution of binding specificity, and we've looked at a narrow subset within the larger solute binding superfamily, which bind amino acids. You can see here the tree. We have glutamine binding proteins here, arginine binding proteins, aspartic acid, glutamate. Because of the short amount of time in this talk, I'll focus primarily here on these nodes here, which is the common ancestor of the glutamine binding proteins and the common ancestor of the glutamine and arginine binding proteins. So these were reconstructed with good statistical support. You can see here in this logo view, most of the amino acids are unambiguously assigned. There are a few that are ambiguous, and these are pretty easy to test. You just synthesize different genes with different amino acids at these positions to investigate the effect of the different substitutions. So looking at these, what we found is, and this is the extant glutamine binding protein, you can see it has affinity for glutamine. This is ITC experiments, so Ka is a measure of the affinity, so higher is higher affinity. So glutamine is specific for glutamine, but it has some promiscuous affinity for arginine, which is interesting. These cationic binding proteins have specificity for cationic amino acids and no affinity for glutamine, whereas these ancestors, QR and Q, they look very similar and they have greater affinity for arginine than glutamine. So something's happened through evolution for them to specialize for glutamine binding in this branch or cationic amino acid binding in this branch, and we wanted to understand what exactly has happened here. So what we saw when we looked at crystal structures of these proteins was if we co-crystallized with arginine, we got a structure like this, which looks pretty much exactly like a contemporary extant arginine binding protein. If we did the same thing with glutamine, we got a very different structure that looked pretty much exactly like an extant glutamine binding protein. And when we looked at the structure of the arginine binding protein, we actually saw some residual electron density which matched this glutamine-like structure. So this shows that these two structures are interconverting even within the same crystal, and that the protein can adopt very different conformations to bind different ligands. So this is quite nice, and what it sort of implies is that there's this conformational plasticity of the protein, its ability to bind to different ligands using different conformational substrates, that underpins its promiscuity or its ability to bind multiple ligands. It's not that you've got one binding site like a lock that only the arginine one fits into well, and the amino acid glutamine may not fit into as well, and this is why the affinity is different. The affinity isn't different because it can't bind them. The affinity is different because these two states have different energies, and this is sampled more frequently. So what this implies is that evolution of this protein family can proceed via conformational selection. So the conformers that can bind all the different ligands, in this case, pre-existed in the ancestor. And so evolution can then proceed by enriching and stabilizing one over another. And it allows a much more gradual and malleable view of evolution, especially within this family, which has evolved to bind so many different ligands. So we then want to look at the emergence of catalytic activity from this fold. And 95% of the proteins within this fold are binding proteins, but there's one clade which is interesting in that they're actually enzymes. So the cyclohexadiene dehydratases, they catalyze the conversion of prephenate to phenylpyruvate. They have very similar structures to the binding protein. So grey is the enzyme, magenta is an amino acid binding protein. You can see that they're extremely similar, but they have no overlapping functions. So the enzyme doesn't bind amino acids, the amino acid binding proteins don't catalyze any reactions. So there's a complete separation here of their functions. So something's happened through evolution to allow an amino acid binding protein to become an enzyme, and we wanted to understand what that was. So again, we inferred a phylogeny from the sequence alignment, we reconstructed these ancestors. Ancestor 1, WS02079, it being its extant representative. These are both amino acid binding proteins and are essentially identical in terms of their properties. Ancestor 2, PU1068, we're sure it's a binding protein. It probably binds some type of carboxylic acid. Its genomic context indicates it's a binding protein, but we don't know its native ligand, and this is partly because it's from an oceanic bacteria which are poorly understood. But ancestors 345 and the extant representatives EA1174 and PACDT, these are all enzymes. So here's an example here. We knocked out the pathway in E. coli, and it can be complemented and reconstituted with expression of these enzymes 3, 4, and 5, but not with CDT1 and 2, so the earlier ancestors. So something's happened between ancestor 2 and ancestor 3, which has introduced enzymatic activity to the fold. So we know, looking at the extant CDTs, that the catalytic amino acid is glutamine 173. This is the catalytic general acid. Is it that simple? It's not, because when we look at ancestor 2, which we know isn't an enzyme, this already has this catalytic acid here. And in fact, if you look even closer, the active sites are basically identical in terms of their sequence. So there's something else going on other than just having the right amino acids in the active site. So we performed a directed evolution experiment. So this is, again, a synthetic biology technique where we mutate randomly throughout the gene and identify for mutations that improve functions. And what this showed is that with as few as 6 mutations, we can introduce activity into ancestor 2. But every mutation was remote from the active site. Not a single one of these mutations changed the active site or the binding site itself. What we found that they did is that rather than change active site, they stabilized the optimum active site geometry. So a really similar scenario to what we saw in the previous study with how the binding site was conformationally plastic and different sites could be enriched through remote mutations. We see the same thing here. The remote mutations seem to enrich different conformations that the binding site can sample. So evolution required glutamate 173 to be incorporated in ancestor 2, and we presume that is for the orphan function. So it's consistent with it binding carboxylic acids. And this evolutionary change, which was under selection, which allowed it to bind its new ligand, then allowed the catalytic activity to emerge with these additional mutations surrounding it. So the other interesting thing that we want to look at here is that from CDT3 onwards, the active site is basically identical, but the activity is very low for CDT3 compared to PACDT, the native protein. So what else is contributing? You can see here the kcat over km increases from 5.6 to 10 times 10 to the 5, even though the active sites are basically the same. So what's going on here? The important things to note here is that there's a big jump in terms of catalytic activity from ancestor 3 to ancestor 5, and there's a big jump in terms of affinity or decrease in Km from ancestor 5 to CDT. So we can see here that the increase in kcat between ancestor 3 and ancestor 5 is probably due to this mutation in the second shell, tyrosine 117 to glutamine, and what this does, this allows phenylalanine 156 within the binding site and glutamate 173, which we know is a catalytic amino acid, to adopt a better conformation. So this explains the jump in activity, or kcat. But if you look at CDT5 compared to the native CDT, they have identical active sites, both in terms of sequence and structure, but a 10-fold difference in Km. And all of the changes in these are remote. So what's going on here? We use a technique called DEER, which is an EPR-based technique, and this reveals distinct conformational sampling in solution across the evolutionary trajectory. So CDT1 is open-closed. If you add ligand, it goes to the closed. This is classical, it's known for the fold. CDT3 and 5, though, they sample as wide-open conformation, and we think this probably relates to as a sort of a vestigial trait from whatever was required for ancestor 2 to bind its orphan ligand. CDT3, on the other hand, has a broad distribution around a closed state, which again is consistent with an enzyme. So despite identical active sites, we see really big differences in dynamics. And this, we assume, underpins a big change in Km. So when we look at this with MD to complement the crystallography and the DR that we did, so the crystal structures are shown here in grey if they're from this exact enzyme, and white if they're from related enzymes, or from the MD simulation. And what you can see is CDT1 open closed and some sampling of this wide open state. CDT3 and CDT5, they predominantly sample this wide open state with a little bit of sampling of open and closed shown here. But PA-CDT, it's mostly a broad distribution between open and closed shown here. So the key thing here is that this wide open state, which has no value for the enzyme, it's not catalytic at all. It has low affinity for the ligand and it can't catalyze the reaction. This state, which is useless for the enzyme, is through evolution essentially frozen out or eliminated, going from CDT5 to CDT, and this corresponds with a 10-fold, 20-fold reduction in the Km, which is much better in terms of ligand affinity. So how did this happen? What we see when we look at the structures is, interestingly, CDT, the wild-type enzyme, forms a trimer. And this trimer has an extended C-terminus. This provides additional C-terminal stabilizing interactions for the closed conformation. The trimer itself prevents it extending into the wide open conformation, and the flexible hinge region is altered to disfavor the open state. So in summary, what we found through this work, which was, like I said, underpinned by gene synthesis, was high energy, rarely sampled states can be stabilized and enriched through evolution to drive functional divergence. For the emergence of catalytic activity, the active site and the binding site residues aren't sufficient. You need remote mutations to stabilize them in the best orientation. And then optimization of catalytic activity from low-level promiscuous activities to truly native-like catalytic efficiency requires optimization not just of the catalytic active site, but of the full conformational landscape of the protein. So Nobu Tokariki helped us out with the directed evolution, Gottfried Odding, Daniela Goldfarb with the DER experiments, and Ben and Joe were the key researchers in my lab for these projects. I'd also like to thank the various funding agencies. So thanks very much, and I'm happy to answer any questions.